So aligning the significance of the alignment scores or doing some statistical test in order to get those scores, either they are correlated with one another in nature or it's just a chance event, that is an important aspect which should have been taken with very careful estimation. So assessing the quality of MSA is an important step in multiple sequence alignment. And what we do over here is sometimes we try to do some randomizations. So we try to do randomization or Monte Carlo test of significances, where we can come up with some group of solutions and we try to correlate some statistics with those solutions and try to pick up which are statistically feasible tests. So in order to do that, we do those randomizations. This is an example where I've tried to show you that how these randomizations are done. For example, we take these columns 1, 2, 3, 4, 5, 6, 7, 8 and 9, whatever columns are. What we do over here is we take the whole columns and we shuffle them, and in this way we can generate many different alignments. So that is we can create some randomization. Same way, within those individual sequences we can shuffle the nucleotides or move the nucleotides and change their positions. So we take the sequences and align them, and the score of the alignment is recorded, and then we shuffle the sequences such that they maintain their length and amino acid composition, but we do that randomizations, and then we compare the shuffled sequences and calculate their scores. Then shuffling and realigning process is repeated. Different steps, or some people they say that you have to do that 100 times, and then this technique is we will apply in our trees, also our phylogenetic trees. We call it as bootstrapping over there. So something similar we are doing here. So we calculate the mean and the standard deviations for those scores. So we have the scores, we have many scores. In this way we have done many randomizations, so each time we came up with some scores. So obviously we came up with some distribution of scores. So whenever we have a distribution, we can calculate its mean and we can calculate its standard deviation. So what we do over here is then we can come up with some Z scores, and then we take our alignment, the real alignment, and see that where it exists in the curve of these scores, while we are taking the mean of randomized alignments in the middle of this distribution. So we do that hypothesis testing. Let's briefly revise what is z-score. So z-score is number of standard deviations, or r-score is below or above the mean, and it can also be called as a transformed score or standardized score. It will have a mean of 0 and standard deviation of 1. That's what the z distribution looks like. Looks like this. We have seen that earlier also, but my apologies guys, it's not a statistics course, but statistics involved. So I try to capture some major concepts and bring it in front of you, but I know they are hard to understand, because for that you probably need to have a full statistics course. But let's look into this z-score one more time. So z is your standard normal curve, or a curve that represents a normal distribution. So let me revise what a normal distribution is. A distribution that looks like this, perfectly bell shaped, where you have a symmetry, right and left, they are exactly equal. This curve is centered around a mean, which is a mean of 0. And then we have 1, 2 and 3, which is actually our z-score, and same way towards the left we have -1 and -2. This is, one is z-score where something is one standard deviation away from your mean. So since your mean in this example is 0, if we look into this formula, this big mean is this mu right here. So that is your mu 0. Since mu is 0, something which is one standard deviation away over here, if we put 1, this sigma is equal to 1, so your answer is y, which is actually in this case it becomes right here. So one standard deviation away from the mean. If your mean is 0, your z value is 1, for an example where you 1 standard deviation away from your mean. A z of 2 means you are 2 standard deviation away from the mean towards right, and then a z-score of -1 is 1 standard deviation away from the mean towards left. Standard deviation is given by squared deviations from the mean divided by total number of observations. So we have seen that back in some statistics primer lectures. Same way we can come up with some probabilities. So here 2% of the values are somewhere here. 0.02 is actually 2%, and 0.15 exactly 50% of the values are towards left and 50% toward right. So your p is 0.5 at that point, probability of seeing a number in this curve. Whereas probability of seeing a number in this curve over here is 0.98. So we can say that number is actually present in upper 2%. If you take the whole distribution and you take those extremes, within these extremes are the tails, you can find very weird event, very significant events, the events which may not be part of this distribution. So they are really different from this distribution. So referring to our alignments, we can say that z-score over here is number of standard deviations by which the real alignment score exceeds the mean of scores for randomized alignments. So there is a threshold which was given by some scientists. If that z-score is greater than 6, then the real alignment score is significantly different from the random alignment scores. So they say it is highly likely that the two sequences are related if their z-score is greater than 6. So let's elaborate that with this example. So for example, we do those randomizations, we came up with distribution of random scores, and we came up with their mean. So this mu is the mean which we get it from those randomized alignments. So that is, for example, we came up with a number that is say 3. We came up with a number for the real alignment, so that is, we can call it as y, or right now we can call it as y. So we can have that y is equal to 10, and let's suppose the sigma or standard deviation here in this example is 1 in order to have a simplicity, or we can come up with some simple calculations. So we draw a distribution that is centered around this mean, where we have this in the middle, we have this mean 3. Now we want to see how many standard deviations away this y is from this mu. So when we do that calculation for that z-score, we see that z is equal to 10 minus 3 divided by 1, so that is equal to 7. So let's see where this 7 lies on, z of 7 lies on this scale. So if we draw the z scale also in parallel, we say that this 3 in this example, if we take 3 in this, we can have a z-score of 0. This is our z-scale. So z-scale goes like this, 1, 2 onwards. So this 7 somewhere lies here in this tail region. It's a really big z-score. So we can say that it's 7 standard deviations away from this mean, this score of 10 actually, which is the score of the real alignment. So this is, since its z is greater than 6, its z we calculated was 7 actually. So that is, it lies somewhere here. So it means it's a very significant event. Probability of finding this event is really low. So since we have the p-values also going in this direction where we have these z's, so in this way we can say that these sequences are very related with one another, and this comparison we have performed with is between the real sequences versus a distribution which we get from the random sequences. So though z-score is a powerful measure, but it does not help to locate the sequence alignments which are incorrect. So we can come up with these scores, and since we are playing with the distribution of the scores, but what's actually there in the real alignments, or what's actually the situation between those two or many nucleotides which has been aligned, that it does not pick. So we have different tools for multiple sequence alignments. There is Clustal Omega, Clustal W, we have DB Clustal, we have K-Align, Mapped, Muscle, MView, T-Coffee, WebRank. These ones, one of them we will just look into in this course, but you will do rest in your homeworks, some of them. So we see that multiple sequence alignment programs will align the sequences, any sequences with one another, and it's in order to have their reliability, it's good to have their statistical testing where we can have some criterion for reliable alignments.